Hello, everyone. My name is Andrew John Cruz, and this is going to be my presentation for Biology 315, otherwise known as cell biology. And this is going to be my honors in the major project for this class. The topic of discussion is going to be myotonic dystrophy. So what is myotonic dystrophy? It is a type of muscular dystrophy, and it's pretty much seen by progressive muscle weakness, especially with skeletal muscle. And it's also known to cause some damage to smooth muscle, and it's usually the inability to be able to relax muscles or after contraction. So this is why it's known as myotonia, and that myotonia is the inability to relax your muscles even after vigorous efforts to do so after using them. So when we're considering myotonic dystrophy, there's two types. There's myotonic dystrophy 1, which is the more severe case. There's also myotonic dystrophy 2, and that's the less severe case. And I'll be going into these a little bit later in the presentation, definitely more in depth. And this disease usually affects around 1 in 8,000 people around the world. So if you think about the entire population, this does affect quite a bit of people in every nation. So if we look at this diagram that I've posted here, that myotonic dystrophy 1 and 2 differ in certain places. Myotonic dystrophy 1, these affect usually the distal areas, which are the farther from points of attachment areas in the lower extremities, as well as the distal muscles in the upper extremities, so like in our hands. So all the red places that are really labeled on this diagram. They also affect the muscles in the face as well as in the neck, as can be seen in the diagram. In myotonic dystrophy 2, these affect more of the proximal muscles in the upper as well as the lower extremities. So these are closer to points of attachments in our body, and as well as neck, the shoulder area, the diaphragm, things as such. And there definitely may be overlap between the two diseases. It's still quite unknown there's still a lot unknown about each two, but we'll talk that more into the later the presentation. So how do I get myotonic dystrophy? So usually it's not something you catch, so you can't get it from being airborne. You can't get it from like blood transfusions or anything like that. So this is purely genetics. And it's the passage of it is autosomal dominant for both diseases, myotonic dystrophy 1 and myotonic dystrophy 2, also labeled as DM1 and DM2. And it exhibits anticipation. So what I mean by anticipation is that when we pass this disease from generation to generation, the disease can come usually earlier in your life and usually more severe when passed down from generation to generation. So if we look at this pattern of autosomal inheritance, if you at least need one parent to have it, and that one parent who has it has the dominant trait. So all you need is one bad copy from one of your parents, and there's a very big possibility you can have it. So if for example, if your dad has a condition, then there's a 50% chance, the children have a condition, knowing that the mom doesn't have the condition. And that's just simple genetics. So, but to have it, you at least need one of your parents to have it. So more into myotonic dystrophy 1. So myotonic dystrophy 1 is caused by something called trinucleotide repeats of nucleotides CUG in the 3' untranslated region of mRNA transcripts of the DMPK gene on chromosome 19. And this DMPK gene is known as myotonic dystrophy protein kinase. So it has a lot to do with myotonic dystrophy. So the usual person will have 5 to 38 repeats in the CUG in the 3' UTR of these DMPK untranslated region genes, but the infected people who have this disease have, the threshold of having the disease is 50 to, definitely thousands of these copies of these repeats of CUG repeats many times. So it doesn't mean that the protein has the mutation. The protein doesn't have the mutation. It's in the untranslated region of mRNA prior to translation. And knowing this class, it's taught a lot about the regulation of translation and transcription and such. So it definitely plays a role in how this all works. So like I said before, the specific phenotypes include muscle weakness and wasting. Definitely cataract development. That's not seen in every patient, as well as heart conduction problems, which definitely makes sense as this does affect smooth muscle, cardiac muscle, and such. So the DMPK gene, the myotonic dystrophy protein kinase gene, it's definitely not well studied, but they do think it has something to do with regulating actin in the cytoskeleton. So this has a big role in how muscle stability plays and how strong our muscles are, muscle fiber maintenance, and calcium homeostasis, although the function's lost. So the increased CUG repeats in these 3' ETRs is definitely a sequestering site for this MBNL type protein. So MBNL is a type of protein that usually is involved in alternative splicing of different proteins. Although when we have this disease, it definitely gets sequestered in the CUG repeat. So it's almost like it's an inhibitor for it because it takes it away from its normal activity and brings it to the CUG repeat in the DMPK gene. So in the nucleus, if the CUG repeats occur in the 3' UTR, they can form this hairpin structure, which I'll show you in a bit. And these MBNL proteins can bind to them and not be able to diffuse out of the nucleus fast. And eventually they'll form these aggregates, which are protein these clumps together. And this is all due to that slippage of the CUG repeats. And it's hard if it's hard to get out of the nucleus. It's hard to do translation and stuff like that. Being sequestered is also another thing. So this stimulates the disrupted splicing in the CTNT gene and the IR gene. So CTNT is the cardiac muscle troponin 2 gene, and this usually regulates muscle interactions and leads to different cardiomyopathy if it's messed up. It also disrupts splicing in IR, and IR is the insulin receptor gene, and we'll go into that a little bit later as well. So the other major protein in DM1 that has the biggest effect in the disease is CUG-BP1. And CUG-BP1 is a protein that will bind to CUG repeats, but not on double-stranded CUG repeats. And it doesn't usually co-localize with the RNA, so it's not really localized there. And it is not sequestered like MBNL1. So these two things add together, the sequestering of MBNL1 and CUG-BP1 now free can now give alternative transcripts to people with this disease. So here's kind of the process I was talking about in the last few slides. So if we have MBNL1 being sequestered, here we have this hairpin loop forming. Now that this is double-stranded kind of RNA to think about, this is now a site that MBNL1 can bind. And if you kind of see that MBNL1 is usually for alternative splicing of other genes, and now that this CUG repeat in the 3' ETR of this gene takes it away from its normal activities, it can't usually do what it does. So this causes problems in certain genes that we'll talk about in the next slide. Similarly, CUG-BP1 usually will bind to the CUG single-stranded RNA in the 3' UTR. So in normal people, the 3' UTR has CUG repeats around 5 to 38 of them, and it can bind there in the single-stranded state of normal people. But when they form this hairpin loop here with these many repeats, now CUG-BP1 doesn't bind to these double-stranded RNA sites. And with protein kinase C activation, CUG-BP1 can be increasingly stable and can do things like affect alternative splicing, certain mRNA translation, and can cause decay of this mRNA. So if we look here, usually MBNL1 has a big case in these three genes: IR, CIC1, and TNT. So CTNT again is cardiac muscle troponin 2. CLC1 is these chloride channel genes, and IR is the insulin receptor gene. So usually, as you can see, they have different alternative transcripts depending on whether you have the MBNL1 doing its normal job or in the disease-ridden people where MBNL1 is sequestered and CUG-BP1 is now free. So if CUG-BP1 is now free, it gives a different alternative transcript to this IR gene. So now it doesn't have exon 11. And without this exon 11, this leads to insulin resistance in some patients. So, something like diabetes, type 2 is now more prevalent in these people. And, that's a whole nother story. The next thing is in CIC1, CGBP1 can now have a premature stop codon in this CIC1, like I said, the chloride channel genes. And as we know in earlier classes, these chloride channels are really important for neurotransmitters as well as muscle relaxation and flexing. It's important to get the information in and flex our muscles. But now if you have this premature stop codon that CGBP1 has introduced that should have been spliced out, this can lead to myotonia, like which I said before is an inability to relax your muscles after hard topics to relax them. Lastly, the CTNT5 can have an alternative transcript with CGBP1 with an extra exon 5 here, and this can lead to, like I said, different cardiomyopathies. So the next type of myotonic dystrophy is type 2, and this is usually responsible for around 2% of the cases of myotonic dystrophy. So DM1 exhibits around 98% of the cases here. And the primary cause is these CCTG repeats in the CNBP-ZNF9 gene, which is located in chromosome 3, and it's in the intron region. And it can have a lot of repeats in this region as well. So introns, they're not coding DNA, but if the damage is long enough, these can go into exon regions and such. And more specific phenotypes of this, like I said, are early onset cataracts. Again, not every patient, muscle weakness, myotonia, and other overlaps with DM1. In it's very similar to DM1. MBNL1 is sequestered in DM2, so it follows the same kind of mechanism and sequence as it does. Usually the biggest difference between the two is that the CGBP1 is not really seen to be overexpressed in DM2, so that last part of CGBP1 not being free, it's not seen. So this gives rise to MBNL1 being sequestered, and the primary cause of the abnormalities in DM2, and it's usually not as serious as DM1. So the reason that this CGBP1 is now not as active as it was in DM1, this is really the reason why it's not as severe. So the major cause of the disease is MBNL1 being sequestered and not being able to give the proper alternative transcripts. So that's why they have less serious muscle wasting in relation to these splicing abnormalities. And as with everything, it begins to be increase in seriousness with age. So a very common of these myotonic dystrophies, muscular dystrophies, is this regeneration, degeneration kind of theme. So now that your muscles, are weaker because of this, actin not being in the cytoskeleton, not being as, strong as it should be, having myotrauma, being able to break these muscle cells is way easier. So we're having these, proliferation of these muscle cells, and with constant degeneration and regeneration, we have centrally nucleated nuclei, which is not really normal for regular cells. They're usually on the outside, as you can kind of see here. And having a lot of these centrally located nuclei is a sign of these dystrophies, because we're able to degenerate the muscle cells very easy, and then now we make more, and eventually we're going to run out of these muscle cells to regenerate. So currently there is no cure for myotonic dystrophy, which is not that great. Usually what they do is they treat all the the various traits, such as the muscle weaknesses, stuff like that, but there is no overarching cure. But there is research going on for cures. So some of the things they're doing in terms of research is this antisense drug therapy. So what can happen here is we have this antisense oligonucleotide. So they're these short strands of like complementary mRNA to the mRNA, where they can bind there and physically block translation from happening, so that we don't get these proteins such as CGBP1 being so toxic to the body, and this can physically block translation, or it can cause it can get things like RNase H to come over and degrade the mRNA, stuff like that. The biggest thing here is to just degrade the mRNA that creates the protein, such as CGBP1. And the other kind of therapy they're having is someone by the name of Matthew Disney, to free the protein such as MBNL1 to create these small molecules that can free the proteins like MB and L1 so that they can go back to doing their normal thing. So I think with, a little bit of both, being able to, stop the protein synthesis of CGBP1 and being able to free MBNL1, this could be very promising for the future of myotonic dystrophy and such. So the conclusion, myotonic dystrophy is, very toxic and, increases in severity with age. But, hopefully with the right treatments that are being researched, we can, have a lot of these things to be, not worry about as much. And myotonic dystrophy, with its muscle weakness, muscle waste, can be a thing of the past. Thank you for watching. My name is Andrew John Cruz, and this is my Biology 315 honors in the major project. And that'll be all. Thank you so much.